forms, either oxyhemoglobin or deoxyhemoglobin. So oxyhemoglobin, this is when the iron shares the electrons and is able then to bind with oxygen. Deoxyhemoglobin is the reduced form of hemoglobin, and so this is when the oxyhemoglobin has dissociated and released oxygen and the heme iron is still in the reduced form. The hemoglobin does not lose an electron when it combines with oxygen and is not oxidized. Another form is carboxyhemoglobin, and this is a reduced heme that combines with carbonic monoxide. And this can be very dangerous because carbon monoxide is 210 times stronger, and it binds where it binds at the oxygen binding site on hemoglobin rather than oxygen binding to that site. Okay, and so this is going to decrease the amount of oxygen that combines with hemoglobin in the pulmonary capillaries. So people with carbon monoxide poisoning don't show any reflex to increase ventilation, and thus why it is very dangerous when we have exposure to high levels of carbon monoxide. The oxygen carrying capacity of blood will be determined by the concentration of hemoglobin. And so this is the maximum capacity of hemoglobin to bind to oxygen. And so some conditions can occur. So anemia is one of them, where we have concentration levels of hemoglobin that are very low. Also, we have polycythemia, which is the opposite, and it has high levels of hemoglobin that are above normal due to an increase in red blood cell count. So hemoglobin production will be also controlled by erythropoietin, which is stimulated by oxygen delivery to the kidneys when it's low. And the production will then be stimulated by increased levels of carbon dioxide. And so hemoglobin will be affected by either loading or unloading. And so loading is when hemoglobin will bind to oxygen in the lungs, and then unloading is when oxyhemoglobin drops off oxygen to the tissues. Okay, so loading is going to occur within the lungs, and unloading will occur within the systemic capillaries. So factors that can influence loading and unloading will be partial pressure of oxygen within the environment, where when we have an increase in load, we'll have an increase in oxyhemoglobin. Also, the affinity or the bond strength between hemoglobin and oxygen also will determine how much loading and unloading happens. And so this is going to be a direct relationship that depends on oxygen of the environment and the affinity for oxygen. So high levels of oxygen will favor loading, and then also strong bonds will favor loading and inhibit unloading. The effect of the partial pressure of oxygen on hemoglobin saturation is determined by the oxyhemoglobin dissociation curve. All right. So with oxygen unloading within the systemic arteries, these will have a partial pressure of oxygen at about 100 millimeters of mercury. So this is termed at 97% saturated with oxygen on that oxyhemoglobin, which equates to about 20 milliliters of oxygen per 100 milliliters of blood. So with the systemic arteries, this goes to capillaries so that the oxygen goes to the cells and for aerobic respiration. That's why we have a high content of oxygen. While within the systemic veins, though, we have a partial pressure of oxygen that decreases down to 40 millimeters of mercury. So this is now 75% saturated with oxygen on that oxyhemoglobin. And this equates to 15.5 milliliters of oxygen to 100 milliliters of blood. And so 22% of our oxygen is unloaded in our tissues, which is about 4.5 milliliters of oxygen per 100 milliliters of blood that is unloaded. And so what we have here is a graphic illustration of percent of oxyhemoglobin that's saturated at different levels of partial pressure of oxygen. Okay, so here on the graph then what we have is percent of oxyhemoglobin saturation and then partial pressure of oxygen on the x-axis. And then here is the volume of oxygen content per 100 milliliters of blood. And so this graph is used to predict what unloading percent will be at any given time. So with this graph, what we find is that within the arteries, we are at about 97% saturated, okay? Whereas with the systemic veins, then we go down to about 75%. And so this graph has this S shape to it, or sigmoidal shape, and this is due to high partial pressure of oxygen, and the changes in oxygen levels have little effect in loading. So at the steep part of the curve, what we have are small changes in oxygen produce large changes in percent saturation. And so here I'll use another color. So here we go. Here are those steep levels right here. So changes, again, small changes in partial pressure then affect percent change in saturation. And so when we also have oxygen remaining in the veins as reservoirs, so to prevent this from occurring. So if a person stops breathing, there's an oxygen reserve that will keep the brain and heart alive for 4 to 5 minutes without CPR. So clinically relevant would be with exercise too, where oxygen unloading during exercise is even greater. So it's about 22% at rest, 39% with light exercise, but then 80% with heavy exercise. An increase in temperature will also affect the unloading capacity and has the same effect as a decrease in pH. So an increased temperature has the same effect as a decrease in pH, all right? And so the affinity is going to decrease at lower pH and increase at a higher pH. And this is known as the Bohr effect. All right, so if we look at here at 7.4, this middle curve, this is representing a normal physiological condition, okay? So when we have a lower pH at 7.2, this is termed a right shift, all right? And so here we have more unloading that occurs at this lower pH because here we have percent of hemoglobin saturation, and then here now we on the y-axis, and then partial pressure of oxygen on the x-axis, all right? So one way that we would have a lower pH would be as if we have an increase in metabolism and thus an increase in carbon dioxide levels. And so we'd have more oxygen that's unloaded to combat that increase in carbon dioxide, and so this shifts the whole curve to the right. Whereas if we have now an increase in pH, this is termed a left shift, and so we'll have an increase in oxygen and in hemoglobin affinity. We'll have an increase in loading in the lungs, and then we'll have, though, a decrease in unloading of the lungs, or in the tissues. Thus, we'll have a decrease of oxygen within the tissues. So another variable that can affect the dissociation curve of oxyhemoglobin will be 2,3-diphosphoglyceric acid, or 2,3-DPG, and this will affect oxygen transport as well. And if we recall, red blood cells obtain energy from anaerobic mechanisms of glucose because red blood cells do not have a nucleus or mitochondria. And so the response then is to produce this 2,3-DGP. And so one of the roles that the DPG has is to inhibit oxyhemoglobin. And what that does then is increased concentrations will decrease the affinity between hemoglobin and oxygen. And so this will shift that dissociation curve to the right. And this is typically what we see with anemic patients because they have a low concentration of hemoglobin, and so they have to combat that by producing the 2,3-DPG. So fetal hemoglobin, or hemoglobin F, has 2 gamma chains instead of the 2 beta chains, and hemoglobin F cannot bind to the 2,3-DPG and thus has a higher affinity for oxygen. Sickle cell anemia has hemoglobin S, which differs in one amino acid that's inherited, and so what happens is that there's this paracrystalline gel that's formed within the red blood cells due to different crosslinks. And so this makes the red blood cell less flexible and more fragile. And so they has a decrease in ability to pass through narrow vessels and organs. So all of these then are to help promote unloading of oxygen to tissues so that we have that oxygen to go where it needs to go. Another variable that affects the amount of oxygen that's unloaded in tissues is muscle myoglobin. And this is a red pigment found on striated muscles such as skeletal muscle and in cardiac muscle, and these are rich with hemoglobin or myoglobin. The difference between myoglobin and hemoglobin is that myoglobin has 1 heme instead of 4 hemes like hemoglobin does. There will be a higher affinity, however, for oxygen than for hemoglobin. And so this acts as a go-between to transfer oxygen from the blood to the mitochondria within muscle cells. So the partial pressure of oxygen in the mitochondria is very low because oxygen is incorporated into water. And this also might serve a role in oxygen storage function for cardiac muscle, where in diastole, myoglobin loads with oxygen, but then in systole, the oxygen is released. And so here in this image, what we have is the percent of oxygen saturation that's on the y-axis, and then the partial pressure of oxygen on the x-axis. And so with myoglobin here, what we see is this left shift, where up here with the venous blood, we see that it retains most of the oxygen as compared to the arterial blood. So the percent of saturation is at a higher amount than what we see with hemoglobin, which hemoglobin is in this line here, where if you remember, it went from 97% down to 75%, while myoglobin stays at about 99% and then drops down to about 93%.